You hear some bells in the background. I'm actually in Bologna right now. Came here to visit some friends. Okay, so we'll start. Yeah, this is our monthly CASP special interest group on ensembles of conformations. And I usually start with the same slide. And I'm actually not sure who to acknowledge for this slide. I have this citation here. But giving the idea that proteins are not static structures, or nucleic acids for that matter, they sample multiple conformational states, and these are fundamental to the way that they work. And so the CASP, of course, has been focused traditionally on modeling or predicting the structure of the protein or the nucleic acid. And we still continue to have that challenge with certain kinds of targets and complexes. The recent advances in machine learning have addressed a lot of the basic problems that CASP was trying to explore, but there are still many more complicated systems. But this opens new areas, and one new area is can we as a field reliably model multiple conformational states, both the structures and the populations of the states? So recognizing that biomolecules do not have static structures, but rather adopt a distribution of conformational states, this special interest group is trying to develop methods to assess how you would assess predictions of ensembles. And is this problem even a feasible problem? Because there are issues that once you have multiple conformations to fit the data, maybe having more conformations always fits the data a little better. So this is a really open research question. And we expect to make some impact, of course, in CASP 16, but we also expect this to be an ongoing activity to help drive the field of multiple conformer modeling. And when we talk about multiple conformers, we can be talking as something as sort of two states, like the classic thinking of the two-state system of adenylate kinase, or wide ranges of conformations, like we have in intrinsically disordered proteins. And there actually is an interesting discussion. It's been a little in the literature. We've written a little bit about it, about how do you define a state? What is the definition? Of course, if you look at the two-state adenylate kinase, then within each state there are multiple conformations, and so that becomes a little bit a matter of definition of the problem as it's being addressed with the appropriate methods. In any case, we have an organizing committee, and I've acknowledged this group before, and they help put together who we should invite and what targets might be for CASP. And we have and I'll say that comments are welcome in the chat, and we're going to be recording those. They'll be available. If you go on this mailing list, monteleonelab@rpi.edu, then we will provide you links to the videos as they become available. And the videos themselves actually are on YouTube, so people can get them there. And it also means that what we're doing in a meeting today to try to teach the community is available to the broader community, even though they might not have been able to make this Zoom session. And just last point here I wanted to make is that the alternate confirmation SIG, we meet first Wednesdays at 11 a.m. And but we also have a general AI special interest group. And I saw a change of time, but I think it's still the same time. There was a new Zoom link that went out, but I think it's the same time, 11 a.m. on Eastern Daylight Time on the second Wednesdays of each month. And we also have an RNA special interest group that Rachel and others organize. And what I have for it is second Tuesdays. And if that's different, Rachel, maybe you could put something into the chat. And I just point out that part of this is directed toward a CASP experiment, and we have CASP 16. We have tentative dates. I don't think we have a venue yet. I expect it will be announced pretty soon. So today, I conferred with other members of our committee, and we invited John Orban to talk about some of his work on multiple conformational states of proteins. John is in the Institute for Bioscience and Biotechnology Research in Maryland, and also in the Department of Chemistry and Chemical Biology at the University of Maryland. And John has really been a pioneer in thinking about this general problem of switching proteins. Like you might think about adenylate kinase, and we've heard a number of talks about methods trying to predict flipping between two conformational states or two or three conformational states, which are predominantly populated. And John has been working on this for, I guess, about 20 years or maybe longer. He tell us more. So John's going to tell us about proteins that switch folds. And then the last part of the meeting, we have some discussion about the CASP 16 targets. And Terry Ois will tell us about particularly this protein A with interdomain linker target. So we'll come to that. But now, John, if you could take over sharing, John Orban will talk to us about proteins that switch folds.
All right. All right, can you all see my screen?
That looks great.
Okay. So thank you for that kind introduction, Guy. And as Guy said, yes, I'm interested in fold switching, and we've been interested in it. I would say it's been at least 20 years. So I maybe just wanted to start by showing this slide here. This is just showing a continuum of protein structure and stability. And so, we do a lot of work on stable proteins, and stable proteins have these deep energy wells. But even stable proteins, what we regard as stable proteins, are quite dynamic. And so that's over this end of the continuum. But over this other end of the continuum, you have IDPs, and they're highly flexible, and they really don't have an energy well to speak of at all. But in between these, so these two areas have been studied quite extensively. But in between these two, you have proteins that switch between different ordered states. And in these different ordered states, they tend to have shallower energy wells that allow alternative states to be energetically more accessible. And it's this group of proteins that I would like to discuss. So perhaps the first examples of fold switching were observed in prion proteins. And so these are proteins that exist in a predominantly alpha helical state in their benign form, but they can then transition to a multimeric beta strand containing state that is thought to be responsible for a range of diseases. And when these proteins were first observed, it raised some questions about, well, is this fold switching property unique to these types of proteins? Does fold switching only occur in disease states, particularly neurodegenerative disease states, or is it a broader phenomenon? And it was only, I guess over the course of years that it became apparent that actually fold switching is a much more common phenomenon. And I'm showing here some examples of naturally occurring fold switches where there's no genetic change between the two structures. The two structures are 100% sequence identical structures, but what's causing the shift between these two states is some environmental factor. And that environmental factor can be ligand binding. Most often it seems to be ligand binding, but it can be other factors as well. It could be redox, it could be pH. And to date, there have been about a dozen or so of these naturally occurring fold switches that have been I would say, pretty well characterized. But a few years ago, it was suggested, there was a computational analysis that suggested that there were considerably more fold switches present. And this was based on incorrect secondary structure prediction compared with the sequence of something that had been solved and was in the PDB. And so the authors of this paper estimated that up to 4% of the proteins in the PDB were actually switchable based on this ambiguous secondary structure prediction. So that raises a question: can you predict and design fold switches? So we know that there's been significant progress in our ability to predict the structures of unique protein folds and to design unique protein folds. But if we really understood the protein folding code, then we should be able to predict and design protein fold switches. And there's been progress in that. There's been recent progress that's very exciting, for example, with AF cluster, in the ability to predict some naturally occurring fold switches. But but designing protein fold switches remains a challenge. And so this is an area that we've been working in, and it would have a number of benefits. One of which, of course, would be it would help to resolve the ambiguity of the sequence structure relationships, which would have an impact on being able to have even more accurate structure predictions. And then there are other benefits that go with that: improved functional annotation, being able to understand how mutations are related to disease, perhaps even getting insights into protein fold evolution. But also, switches might just be useful. They could have useful applications, therapeutic applications, diagnostic applications. And so this is an area, this has been the motivation for our work in this area. So what I'd like to do is just sort of briefly outline some of the work that we've been doing. We've been looking at designing switches between three common folds that occur in the protein universe. This one here is 3-alpha fold, that we refer to as GA, is an albumin binding domain. This S6 protein is an alpha-beta plate fold that binds to ribosomal RNA, but is also seen in many other RNA binding proteins. And this fold here at the bottom here is a beta grasp fold that we refer to as GB, that binds to IgG, but is also has similarities to a ubiquitin-like fold. And so we've developed switches between all three of these proteins. And what I would like to do is focus, I'll spend most of the time talking about switching between this 3-alpha state and this alpha-beta plate fold. And I should also point out that these folds, there isn't any significant sequence identity between them in their parent states. And these two proteins are actually independent domains within a larger protein known as streptococcal protein G. So basically, what I would like to do, the question we're asking here is, to what extent can you embed the sequence for this 3-alpha fold, for example, into this alpha-beta plate? And let me just briefly outline the strategy for doing this. So firstly, what you want to do is thread the 3-alpha sequence onto the alpha-beta plate fold. And you do this with multiple alignments. And then you try to find an alignment where you minimize the most severe steric clashes and I'm showing the alignment that works best here, where the long helix, the longest helix in the N-terminal helix in the 3-alpha fold actually aligns with this long helix in the alpha-beta plate. Well, we tried multiple alignments, but this is the one that worked best. So once you found that alignment, you want to minimize the steric clashes and to try to resolve those steric clashes by designing mutations to relieve the unfavorable contacts. You evaluate the energies of those in silico, and then you make those proteins. And so the idea is that you're going to increase the sequence identity, going from making the GA sequence more S6-like and the S6 sequence more GA-like. And you're ultimately going to try to hybridize these two sequences and try to co-evolve them to see, well, how high in sequence identity can you go while maintaining the parent folds? And so that is the question: how high in identity can you go? And so, to our surprise, when we did this exercise, we found that you could actually embed all of the 3-alpha sequence into the longer S6 sequence, or what we refer to here as A1 for the 3-alpha and SA1 for the alpha-beta plate. And so all 56 amino acids of the 3-alpha sequence could be embedded into the longer SA1 sequence while maintaining the 3-alpha state in the shorter sequence. And in the longer sequence, the NMR structure was an alpha-beta plate. And you can see the NMR spectra of these are quite different. And so these two proteins, you have one helix that is conserved between the two, that is topologically in the same place. But then these two helices here, the red and yellow helices in the 3-alpha structure, form beta strands or parts of beta strands in the alpha-beta plate. So in the context of the longer sequence, this SA1 sequence, this competition between these two states, this competition between these two states creates a critical state. So depending on where you make mutations, you can get quite different outcomes. You can unfold the protein to something that looks quite disordered, or you can simply destabilize the alpha-beta plate, or if you make more severe mutations, you can unmask the alternative state. So now you've gone from something where we consider it to be in protein folding. We've been working for most of my career on 2-state systems, but this breaks that mold completely. And so this is actually a 3-state system. Now, if you make more subtle mutations, then you can actually populate both folds simultaneously. And that's shown in this top NMR spectrum here, this 2-dimensional proton N15 spectrum, where we see two sets of signals. The red labeled signals are due to the 3-alpha state. The blue labeled signals are due to the alpha-beta plate. And in this particular case, we've made a mutation at the periphery of the core from a valine to a threonine, which destabilizes the alpha-beta plate sufficiently that you can now get a population of the 3-alpha state, but it doesn't destabilize it so much that you don't see the alpha-beta plate. And so it has an effect on the alpha-beta plate, but it's in a disordered region of the 3-alpha structure. And so it doesn't have as much of an effect on the 3-alpha structure. So sequences like this have very interesting properties because they exhibit temperature dependence. So if you look at the NMR spectrum at 15 degrees, you can see this region up here. This is glycines that are due to both the 3-alpha state and the alpha-beta plate. And I've just expanded those here in this series of spectra as a function of temperature. And you can see at low temperature, the protein is primarily in a 3-alpha state. The red peaks indicate the 3-alpha fold. But as you raise the temperature, you start to populate an alternative state. And that alternative state is the alpha beta plate fold, such that by the time you get to 30 degrees, you're now more than 90%, maybe even 95% in the alpha beta plate fold. And we've determined NMR structures at both of these temperatures, and you can go between these two temperatures. It's completely reversible, and these proteins are monomeric as judged by light scattering. Another interesting feature is that mutations can alter the temperature dependence of the fold switching. So you can, for example, if you look at different mutations at that residue position, that position 90, if you have, for example, an isoleucine at position 90, that basically renders the unmasking, the fold switching ability of this protein to be almost undetectable. But as you change that residue from an isoleucine to, say, a leucine, you can now detect at low temperature a significant component of the 3-alpha state. So just going from something that's a beta-branched hydrophobic residue to a gamma-branched hydrophobic residue on the periphery of the core of the alpha-beta plate is sufficient to unmask this alternative state. And then as you make other mutations, you can unmask it even further. So this system is, I would say, very delicately balanced. Small changes can lead, small changes in what appear to be small changes in residue type can lead to large actual changes. And because you can do temperature dependent, monitor the equilibrium constant as a function of temperature, you can determine the free energy requirement for going between the 3-alpha and alpha-beta plate forms over that particular temperature range. So the delta G for this process is approximately about 2 to 2.5 kcals per mole between the temperature endpoints. That is to say, it only requires about 2.5 kcals per mole to go from more than 90% 3-alpha to more than 90% alpha-beta plate. And this fold switching event is occurring over a temperature range that is relevant to biology. Another way of casting this, you could recast this in a Van't Hoff equation, where you plot the log of the equilibrium constant versus the reciprocal of the temperature. And that can give you information about the enthalpy for the reaction. The heat absorbed in the reaction going from 3-alpha to alpha-beta plate is around 22 kcals per mole. There's very little entropy component, so this seems to be dominated by an enthalpic component. And you can also look at the kinetics of the fold interconversion. And we can thank Guy for his experiment, his ZZ exchange experiment, that allows us to do this. So, so this experiment, you'd not only observe the backbone amide proton, I'm showing here of G58 in its 3-alpha and alpha-beta plate forms. Those are the red and brown signals, but you can also see the exchange peaks between these two states. And you can look at the time dependence of these exchange peaks. If you plot the peak intensity versus time, you can get the kinetics of the fold interconversion for the forward and the reverse reactions. And so what you can see is actually the forward and reverse reactions interconvert on a pretty slow time scale, on the order of about 1 per second. And so these are slow exchange events. The main point here, though, is that this experiment definitively shows that these systems are in exchange and they're correlated to each other. They're not two independently varying folds that are just happen to be changing. One's going up with temperature as the other one's going down. These are absolutely connected to each other. Now, you can do this experiment at the temperature midpoint, which is 15 degrees for this particular mutant. But you can also do it at other temperatures. And if you do it at other temperatures, you can get a measure of the activation energy from an Arrhenius type analysis. So you can plot the log of the exchange rate versus the reciprocal of the temperature. And what you see is the energy of activation for the 3-alpha to alpha beta is around 22 kcals per mole. And for the reverse reaction is about 2.4 kcals per mole. So this reaction, this forward reaction from 3-alpha to alpha beta certainly seems to be, it's consistent with the Van't Hoff enthalpy, and it seems to indicate that it's dominated by an enthalpic barrier. As to why this is so different is not completely understood. However, we think it may have to do with the fact that the reverse reaction from alpha beta to 3-alpha is actually somewhat akin to a cold denaturation event, because about 40% of the chain goes from being ordered to disordered in this particular reaction. And cold denaturation is an exothermic process. So it's possible that this partial unfolding of the protein as we go from a high temperature to a low temperature may contribute to heat release, and that heat release is helping to offset this energy of activation requirement for this particular step. That's our working hypothesis until a thermodynamicist, a real thermodynamicist, tells me differently. So I said that we've looked at 3 different switches, and they're summarized here. And so I've already talked about this one, and then we've also looked at switches between beta grasp and the alpha beta plate, and 3-alpha and beta grasp. And you can see that these involve fairly large secondary structure changes between the 3-alpha and the beta grasp. Between the beta grasp and alpha beta plate, they don't look that different until you have a closer look and you realize that the N-terminal beta strand in the beta grasp is pointing in an opposite direction to the alpha beta plate. But these 3 switches, when we look at these, they have some common features, and I'd just like to briefly go over those. So if you look, for example, at, if you look at the core residues in the beta grasp, and you map those onto the 3-alpha structure, you notice that they map to solvent exposed positions. And so it seems like there's minimally, the cores are almost not quite mutually exclusive, but there's minimal overlap between the hydrophobic cores. And this enables the folding information for 2 different folds to be encoded into a single polypeptide chain. At least it facilitates that. And this seems to be a feature that is present in all 3 of our switches. Another feature that is common is that disordered regions play an important role in remodeling the structures. So for example, in the 3-alpha to beta grasp structure, the N and C terminal regions are disordered in the 3-alpha state, but they form 2 beta strands, the central beta strands of a 4-stranded beta sheet. And it's similar case for the 3-alpha versus alpha beta plate, and also for the beta grasp versus alpha beta plate. And it isn't always N's that do this. You notice here, for example, there's a large internal loop in one of the structures that forms a secondary structure in the other, in the alternative state. And the same is true here. So what this enables, it gives the protein, it sort of relaxes the relatively tight structural constraint of having to populate 2 folds with 1 polypeptide chain. It gives it some wiggle room, but it also highlights another fact, that the extent of disorder is highly context dependent. A third feature is that for fold switching to occur, the stability profiles have to overlap. Well, there have to be small energy differences at least. It depends on how you do this. But so for example, if we look at the stability profile for the 3-alpha fold, it's shown here in blue. If you look at the stability profile for the alpha beta plate, it's shown here in black. They don't overlap over this temperature range, but when you make the mutation, the V90T mutation that I mentioned earlier, what it does is it destabilizes the alpha beta plate fold such that the stability profiles now do overlap in a temperature range that is relevant to biology. Now, you can bridge this gap with mutations, or or you can bridge it with an environmental trigger. So as long as you have some equilibrium amount of the alternative state, then that is sufficient. It will enable switching from one state to the other. So what I would like to do now is just briefly describe a couple of environmental triggers that we have designed into these types of systems. And the first example is using our 3-alpha to alpha-beta plate. So at low temperature, this equilibrium lies over to the 3-alpha structure. There is an equilibrium amount of the alpha-beta plate. But we can design, if we design cysteine residues and place them in such a way that they're in close proximity in one of the folds, but far away in the other, then in principle, we should be able to design a system that is redox dependent. And so if we do this, and we do this under oxidizing conditions, where these two cysteines are close enough and they can form a disulfide under oxidizing conditions, we get something that we get a protein, the alpha-beta plate, populating at low temperature, where the free energy of forming the disulfide greatly outweighs the free energy requirement for switching. So the energy gain is much greater than the energy loss from switching, which we know to be only 2 to 3 kcals per mole. The free energy for disulfide formation is in the 10s to 20s to even more than that kcals per mole. Now, if we introduce a reducing agent, we should be able to send this back to the 3-alpha state. And so that's exactly what happens. Under reducing conditions, we can go from alpha-beta plate to the 3-alpha fold. So this has analogy to a native redox switch, the chloride ion channel protein, CLIC1, which does exactly this type of thing, and goes from a reduced state where it has a completely different function, a glutathione reductase function, to an oxidized state where it participates in the chloride ion channel. So, so this is an example of a redox-triggered, an engineered, designed redox-triggered fold switch that we're interested in using, actually trying to develop some applications for it. A second example, I guess I should say a third example of an environmental trigger would be ligand binding. And so let me give you an example of that. So ligand binding can also drive fold switching. We know that from natural fold switches. And in our design system, we have this 3-alpha fold that its native function is to bind to albumin. And the beta grasp fold, its native function is to bind to IgG. But when these were engineered, and initially when these were being designed, the binding epitopes for both cognate ligands were introduced into the sequences. And this was done to enable the proteins to be bifunctional so that we could detect switches through binding. So you have a system like this where the 3-alpha state can bind to human serum albumin, but we have some mutants that can populate some of the alternative state. So for example, this mutant in the NMR spectrum, you can see some of the alternative state. It's actually about 95% 3-alpha, and there's a low intensity signals corresponding to the beta grasp. So what we would hypothesize here is, because folding and binding are thermodynamically linked, that if we were to add IgG to this system, we should be able to shift the equilibrium from 3-alpha to beta grasp. And so these large signals due to the 3-alpha should diminish, and these minor signals should now become the major signals. So when we add IgG, that's exactly what happens. We add IgG Fc, so it binds to the Fc domain. You shift the equilibrium over. And so the free energy of binding greatly outweighs the free energy, the energetic cost of switching, which in this case also is about 2 kcals per mole, and the free energy of binding, well, the binding constant for these is greater than 1 micromolar, and actually is probably significantly greater than that. So, so ligand binding can drive fold switching, and so there are multiple ways you can actually switch between these folds, and this is basically an illustration of how we think about fold switching, where when you get to these very high identity mutants, what you have is an equilibrium between a well-populated folded state and then an alternative state that is a more high energy, weakly populated system. And mutations can shift the equilibrium between these states, and they can be mutations. They don't have to be mutations at the same place in the sequence. They can be at different places in the sequence. And in addition to mutations shifting the equilibrium between different states, ligand binding can also shift this equilibrium. So I hope I've convinced you that it's possible to design switches between commonly occurring folds. There are recurring features that are present in these types of fold switches. In particular, diminished stability and flexible regions usually play an important role. Latent binding surfaces become exposed and lead to new binding functions, and this can happen through either a short mutational pathway or through some environmental factor. And it's also that, just to stress that fold switches do provide insight into the sequence structure relationship. I think they have relevance to structure prediction. I think there are some very exciting developments in the ability to predict natural fold switches, and we hope that our data will help in improving algorithms to make them even more reliable for predicting fold switches. And finally, I just want to say that I think that fold switching is going to be a valuable protein design tool that will be useful for developing various applications, biomedical and other types of applications. So with that, I just want to have some people to thank. In particular, I want to thank Phil Bryan. This has been ongoing work that we have been participating in together for decades now. And most of the work that I described, certainly the work on the 3-alpha to alpha beta plate switch, is the work of an extremely bright graduate student in my lab, Saga Solomon. Yeah, I thank you for the opportunity to speak here, and I'm happy to take any questions.
Thank you. Thank you so much, John. That was great. It was very clearly presented, a very exciting area. I want to allow some questions. I have a bunch, but I'm just going to jump in with one that I want to ask about. So when we talk about fold switching, we typically have the case of like an alpha helix goes to beta strand or to beta sheets, and pretty significant structural changes. And now we see there are, as you point out, natural examples of that. But there are many examples where proteins, they flip conformations, but it's not as dramatic. Like many membrane transporters, they'll have an outward-facing state and an inward-facing state. And they haven't unfolded helices, but they've rearranged helices.
Mm-hmm.
Where's the line between fold switching and these kind of conformational changes? Or is there really, is it really the same biophysics? It's just one is more dramatic than the other.
Yeah, I think you've answered your own question. I think, you know, it's kind of an arbitrary line. I think, you know, in a lot of papers I've seen that there's been this push to try to really define fold switches in, you know, there has to be this alpha to beta transition. And I can understand it from the point of view of trying to figure out which ones are the most significant ones. But the reality is, I think what you just described, fold switching, it's really, there's a continuum of fold switches. And what you've described are things that are maybe more subtle, that maybe the die-hard fold switchers would say, oh, that's not fold switching, but it's on the continuum of conformational change. And that's how I would prefer to view it, because the biophysics is the same. There's nothing magic about fold switching.
I think
And so the same biophysical principles apply. It's equilibrium between two different states. And it's just in one of these states, it happens to be, okay, there's a change in the secondary structures.
yeah, maybe these dramatic fold switchers are very good for training and testing, but what we learn from them will be applicable, and I think you made this point, even in much more subtle conformational changes and flipping.
Right.
So there are some questions that were in the chat, and I'll let them ask. So Gabriel Del Rio, do you want to ask your question?
Sure. Thanks. Hello. Very nice.
Hi, Gabriel.
Hi. Hello, John. Nice to learn about your work. I read your PNAS paper before, but I didn't know what you were doing. And that is made me think about the possibility of finding fold switching in multi-domain proteins. And I'm wondering if you're looking at that.
Yes. So our very first fold switch was between the GA and GB domains, which are independent domains in the streptococcal protein G. And so that's an interesting hypothesis. I mean, that's something we've wondered about, is we were able to develop a switch, and the question is, well, does this actually happen in nature? Did one of these domains duplicate and then fold switch in a multi-domain system to develop a second domain with a different function, so that there was no net loss of function? There was this gain of function through fold switching. So it was a gene duplication followed by fold switching. And I don't know the answer, but I think that it's entirely possible. So, when we look at other multi-domain proteins, there are a lot of eukaryotic proteins that have this S6 type fold as one of the components of a multi-domain protein. And they're often RNA binding proteins. And then on top of that, they have other domains that look an awful lot like 3 helical or ubiquitin-like folds. And so I do wonder about that, whether it might be a mechanism for whether fold switching is actually occurring in some of these multi-domain proteins, where you have these independent beads on a string type systems, where some of them have arisen through some gene duplication and fold switching event. But that's just wondering about it. I don't know it.
I see. May I share something, one last thing?
Yeah, please.
So I've been working with peptides and fusing peptide domains for pharmacological purposes, to target a particular cell or perform some particular function. And what I learned, and this is an empirical observation, I haven't quantified this to be rigorous, but my point is that if I embed peptide sequences within another domain of a peptide, I usually get better results in finding the two functions that I'm looking for than if I fuse the two domains, one on the side of the other. I don't know exactly why is that, but by listening to your talk, I was wondering if these two phenomena are in some way related. I mean, the peptides that I'm working on, some of them display some structure, some others do not. But the general observation in making these fusions or embeddings is in general true. If you embed, it's more likely that you will maintain the structure and function of the two domains than if you fuse together the domains. Just a thought, food for thought.
Well, I see Arnie has his hand up, so go ahead, Arnie. You're muted, Arnie.
Yeah, Arnie, you're muted.
Sorry, do you hear me? Yeah.
Yep,
So
I hear you.
I was interested in the way to try to predict, to identify fold switching proteins. And I mean, there's a number of studies using AlphaFold that you show that you can find both states of most proteins, even the fold switching ones, if you add noise somehow. But that is I mean, it's quite time consuming, and I think still is a bit unclear if you might find incorrect states also. So I wonder, do you have any idea of like finding a quick way to screen for potential fold switches, in particular ones that are not changing secondary structure?
I wish I did. No, I don't. But I can come back to your point about AlphaFold though. So that set of switches that I showed, I can go back to it. You can still see my screen, right? So this set of switches here, AlphaFold gets, I think, about half of these right, and AF cluster also gets about half of them right. So I think there's certainly room for improvement.
Yeah. I think there are better methods than AF cluster to do this that are out there. No, but it's, I agree. Maybe you won't get everything.
Did you just say there are better methods than AF cluster?
There are this way of masking that are better than AF cluster. I think that they might get more variation for less sampling.
I'm not sure. I'd be interested.
Contact Björn Wallner. He has worked on that. So there are. I don't know exactly what. I mean, he has shown proteins when there was a big Twitter fight between some people on the way of things that you couldn't find. I think he quite clearly shows that you can find it but you need to run 1,000 times or 500 times. It's a bit heavy, but I think you can do it. But it's too slow for screening. You can't run every protein 1,000 times with AlphaFold and see if you find it. That's too slow.
Right.
So I'm going to move the discussion on a little bit, and it very much relates to what Arnie just asked and this discussion, which is, how can we do some blind testing of the ability to predict? Actually, going back with some of John Orban's data and testing the various methods like AlphaFold sample and others is a very worthwhile thing to do. But of course, having blind data is a whole new level of testing. And I think people know we are in the process of a CASP that includes multiple conformational states. And I just have just point this out, and this is now in the recording. If you want to go back, there are several targets already from this cycle of CASP, which will be assessed as multiple conformational prediction targets. 1214 is already closed, and these other ones are closed soon, and then there'll be maybe 4 or 5 more of these coming out in June and July. So Andre put this together for us. And there also is some activities going on with multiple conformational states of RNA. And then there's another class of targets which are kind of like this, where we have 2 domains connected by a linker, where the idea is to generate experimental data and then ask predictors to try to predict such systems. And this is actually a really new thing for CASP, and it was suggested by Terry Ois and Bruce Donald, that they could actually provide a target for this cycle of CASP. I just called it target 1. It doesn't have a name yet. And I'm gonna, so Bruce and Terry have advanced this. They're collecting data with Susan Sudakawa. And so Terry, do you want to pick up here? What we're recording here will be available for other CASP predictors who are not on the line, but who may want to try these kinds of targets.
Sure, thank you, Guy. Let me start my presentation here. And trying to make it nice and short. Let's see how well we can do that. So the system that Bruce and I have been working on for the last 10 years or so is something we call ZLBTC. I'll show that in a second. And what we set out to do experimentally was to determine the relative position of this two-domain construct, this fragment, this piece of staphylococcal protein A, that is connected by a flexible linker. And so what I want to do today is just introduce that protein a little bit and show the data that tells us that the linker is flexible, and then get on to the specifications that we are planning to present to CASP. So just to introduce the protein quickly, this is staphylococcal protein A. It's a 524 residue protein as expressed, but it's attached on the cell surface of Staph aureus. And the business end of the protein is what we call SPA-N, these 5 almost identical domains that bind not just IgG, but also many other proteins in the host, and it's a significant virulence factor and has a significant impact on Staph aureus infections. Each of these 5 domains consist of this 3-helix bundle structure. And as you can see from the sequence alignments, they're nearly identical in sequence, very similar. And the sequence that's highlighted here in brown, corresponding to the termini in this single domain structure that we're looking at, comprise the linker sequence. It's a 6-residue linker sequence for all the linkers except between domain E and D, which has an insert of 3 amino acids. But this linker sequence is conserved and plays an important role in the CASP challenge that we're describing. In order to understand the structural characteristics of this linker, we've constructed a protein that takes this highly identical sequence and makes it absolutely identical, repeats the B domain sequence 5 times to make a construct that's very similar to full-length N-terminal domain. Excuse me, I have a plumber that just arrived. Back there, can't talk to you and present. Okay, sorry about that. So these residues, we've measured the order parameters for all of the residues in these in this protein. The order parameters can be determined by N15 relaxation experiments, and we find that only the 6-residue linker has significantly lower order parameters than about 0.95, which is quite high. So that's our evidence that the 5 domains are well folded and stable, but their linkers are very flexible. And so we've constructed the ZLBTC construct to represent the BC pair of domains, 2-domain construct. The Z is a slight variation on B domain that's been made by the biotech industry, a couple amino acid substitutions. And the LBT refers to the lanthanide binding tag, which was developed by Adam Barb when he was in Jim Prestegard's lab. And they inserted a lanthanide binding tag in between helix 2 and 3 of B domain or Z domain. And that allows us then to orient the protein in the magnetic field on the basis of slightly aligning the first domain, the ZLBT domain. And the question we asked is, how does domain 2, the C domain, move relative to the ZLBT domain? In other words, what is its relative orientation compared to ZLBT? And that's something that you can determine with residual dipolar coupling experiments. And we have gone ahead and done that. and the results I'm about to show. But before I do, I want to mention some previous X-ray crystallography results that indicate that among the 3 helices, a helix 1 in this 3-helix bundle domain is the one that tends to vary in its alignment relative to the other 2. And this is shown here by collecting several NMR and crystal structures of B domain or similar protein A domains. And what we see is that if we align on helix 2, helix 3 is well aligned, but helix 1 varies quite a bit. And this is rationale for the way we've specified how to define the coordinate system or the coordinate frame of each domain. To make this point, I want to show you also another crystal structure, which is a crystal structure of the double B construct. 2 domains linked by the wild type linker between B and C. And my student, Lindsay Dice, managed to grow crystals of this highly flexible protein. We knew that from our NMR experiments, but she managed to get dimers of these 2 domains linked together. And the structure that she found, when she showed it to me, I said, well, that's very nice, Lindsay, but you know, it's a crystallization artifact. Because clearly the crystals have selected out a particular orientation. And that's just a way to introduce our system for describing the orientation of the 2 domains relative to each other, which has to do with defining a coordinate frame based on the helical core structure, specifically the structure of helix 1 and 2. This is the construct that we'll be talking about. This is the ZLBT domain in the N-terminal position. C domain, we have an NMR structure determined by Adam Barb. For ZLBT, we have a 0.9 angstrom crystal structure for C domain determined by my group. And it's on the basis of those 2 structures that the challenge is not to predict the structure of the individual domains. In fact, in order to compare with our experimental RDC results, those domain coordinates need to be within, you know, half an angstrom of these structures, okay? What's the challenge is to predict how C domain is oriented relative to Z domain. In other words, how those 2 coordinate frames work. And I'll show you that in a second in more detail than the drawing I just showed. We also, so we have published, and I'm about to show you the results for the wild type linker ZLBTC data, but we haven't given the actual quantitative results. All we've published is the figure that shows how these 2 domains are oriented. And so that is still a predictive challenge. And at the same time, we are in the process of collecting RDC data on the Gly-6 linker version of this protein, where we've replaced all of the residues in the wild type linker with 6 glycines. And that we will be determining the relative orientation of the domains. Elaine Napoli will be doing that in the next month or 2. So how do we show these orientations? We're doing it by determining not an ensemble of structures, which is the typical way that RDC analysis is done for flexible proteins, but rather by determining a continuous distribution of interdomain orientation, which we call CDIO. So this is a 3-dimensional normal probability distribution. It's called a Bingham distribution when it's on SO3 in 3 dimensions. And this distribution can be displayed as this disk on sphere representation. And as with many RDC measurements, there's a redundancy in the inability to determine whether a dipolar coupling is resulting from bottom to top or top to bottom. They both give the exact same dipolar coupling, and that results in two different possible solutions. And these solutions are shown here. Now, the way we display this is by showing the orientation of the Z prime axis of the second domain, the C domain, the blue domain. We show that orientation as a particular position as a disk on the sphere. And then we show the orientation of the X prime axis as a probability distribution encoded by this color scale for the position of the X prime axis. This is a so-called axis angle representation, and the best way to compute it is with quaternions. So basically, a quaternion has 4 components. 3 of them define the position of the disk, and the 4th one defines the rotation angle around that. And with that information, we can rotate the coordinate system of the second domain onto the coordinate system of the first domain, and that is a definition of the interdomain orientational distribution. I mean, it is a distribution, as you can see. There's a quite wide range of probabilities. And so the challenge is to predict that the mean and variances and covariances of this probability distribution, which is what we've observed experimentally. Now, you might ask, how do we choose between the two solutions? Actually, our proposal is to compare both solutions with the predictions. So we're agnostic about which prediction, which solution might be correct, but we did our own ensemble simulation, and on that basis, we concluded that solution 2 was the more probable solution. And this is an example of what we will do with an ensemble prediction, which is to kernelize each of the individual conformers weighted by their probability, and then add those kernelized probabilities up to get a CDIO like the ones we're showing in these spheres. So just to very quickly show you how these reference frames are defined, I think you can see this thing I'm rotating now. Is that correct? I hope we're seeing it. Are we?
Yes.
Okay, good. Yep. Yeah, so the ZLBT domain is the so-called reference domain. It's shown here on the top with the black XYZ vectors showing the coordinates of that reference frame. Helix 2 is shown in red on this view, and helix 3 is blue. And because those are the 2 helices that are least likely to vary in their relative orientation, those are our reference helices. And so we make the z-axis of our reference frame parallel to the axes of those 2 helices. Then we, using singular value decomposition, and then we use the single value decomposition to find the bisector between the 2 helices, and that turns out to be the x-axis of our reference frame, and then y is perpendicular to x and z for a right-handed coordinate system. So that's our reference frame, and then we can take the second domain and do the same calculation using singular value decomposition. So here again, the z-axis is parallel to the helical axes, and the x-axis is perpendicular to it, running through helix 1. That's the direction that the x-axis goes. And so now we've defined frames for each of the domains, and the challenge then is just how do we place the red domain in the black frame, the red frame in the black frame, and that's shown here in the magenta. So for this structure, which is that 2B crystal structure that I showed, this is the relative orientation of the 2 domains. And it's clearly based on the CDIO figures I just showed, it's a very low probability structure. So crystals can actually pick out of solution quite low probability structures out of an ensemble of structures. So Guy has copies of the specifications that we've written for this challenge, and they basically describe the algorithm for defining this reference frame and the set of rules that we need to use to, I'm having trouble getting to stop share. The rules that we need to use to accept structures, structural predictions. Now we're hoping that someone will actually try to predict directly the continuous distribution and probability distribution, but we're anticipating that most of the predictions will come as ensembles, and we will apply this algorithm I just showed to generate a CDIO, which we can then compare with our experimental CDIO by measuring the free energy difference between the two distributions. And for more information about that, you can consult the paper that describes this first pass of these two-domain structures.
And so, like I said, there are some predictors on the line here. Many others may listen to this, hopefully. But so the idea is that we want to be able to understand dynamics of an interdomain linker. And these are also affected by the fact that the two kind of balls at the ends can't overlap. And so in order to do this, in your system, the three helices at each end, called the two balls, have to be fixed to the coordinates that you provide. In particular, helices 1 and 2, or 1
2
and 3. 1 and 3. I should know because I actually worked on this system for many years. But in any case, but should they fix the three helices or just two of the three helices?
Just 2 and 3. Well, 2 and 3 need to be within half an angstrom RMSD from the deposited structures, so that the frame that we define agrees, right? So we have to use those frames. In terms of what's going on with helix 1, it isn't necessary for defining the frame, and therefore that's less important to have its RMSD be exactly the same as the deposited structures.
So a predictor would deposit some number of structures and their populations.
Yes.
And the point is that the helices 2 and 3 have to be very close to the deposited NMR and crystal structures.
Right.
Okay.
Otherwise, we can't define the frame, and if we can't define the frame, we can't calculate the CDIO.
So does someone, I see Conan has a question. Conan, do you want to say your question, or is that from the previous talk?
Previous.
Okay.
Oh, well, it's also a new one.
Okay. Does anybody have a question then for Terry or for
I do.
Go ahead, Gabriel.
Hi, Terry.
Hi.
I'm not sure exactly why do you choose to use a 3D system for answering the questions you have. It seems to me that your system can be represented in an adjacency matrix, and that matrix will be diagonalizable. Sorry for my English there, and which means that you can find the eigenvectors, which will give you the base of the transformations, the geometrical transformations that you are looking at.
Sure.
So I'm not sure why you prefer to use the 3D system and to find this particular helix or whatever point of reference you choose for studying the transformation. Can you explain this?
Well, I think even in the case of the density matrix you're talking about, which I agree, that's actually very close to the mathematical representation of the rotation matrix that's necessary to rotate one domain, the C domain, into the Z domain's frame. So that representation could be used instead, but in both cases, we would need to use the coordinate system of the domains themselves in order to describe their orientation relative to each other, right? So it's a first step in the probabilistic description of how they're oriented. But without reference frames, I don't think we could do that, right? Because we have to have some structure-based way of defining what those frames are in order to describe how they're related to each other in a three-dimensional sense. Am I missing something?
Yeah, well, I see your point. And that made me wonder whether let's say if you have two conformations, if the two matrices their eigenvalue vectors will necessarily have the same reference system. That's a good point. I haven't considered that.
Yeah, so I mean, basically the singular value decomposition is really finding the eigenvectors, right? And so that's exactly how we use the coordinates in these domains to define these axes that I showed.
Okay, thank you.
You're muted, Guy.
Thanks. I've been trying to keep these these Zoom meetings to the time, to the hour, but I thought it was important to let Terry have enough time to explain this experiment because it is quite new and it's quite interesting. I guess, Terry, you're also going to get some SACS data, and we're going to also try, if we can, to get a second system that maybe have data available and can be a second CASP target this summer. So does anybody else have any other questions before we wrap up then?
Just to make one quick comment, Guy, it is very important, and I forgot to mention that all of what I said was focused on the interdomain orientation, not the interdomain distance or the translational relationship. We're hoping that by combining our NMR results with the orientation and the SACS results, which should be sensitive both to distance and orientation, that perhaps we can get the true description of how one domain is oriented and located relative to the other. Yeah.
And Terry, if I could add one point that I think might help some of the questions, is that the reason we're being quite particular about defining the coordinate system is so that we can compare fairly the submissions to ground truth. If we didn't do that, then there's the potential that a prediction could be differing by a rotation, which of course is the center of the distribution, which is one of the most important characteristics. So it's actually an interesting point, is that I even have heard in our community, sometimes people say, well, why do you want the optimal solution? Or why do you care if your algorithm is correct? Well, in this case, SVD is computing the optimal alignment frame. And that means that we can fairly compare the ground truth to a predictor's contribution. If we use some method that wasn't computing the best fit frame and being careful about it, then we could still do a comparison, but we might be off by essentially a rotation. And this way we can eliminate that error as part of the judging bias. So that's one reason we're being careful about that, to define it.
When the target's released, it'll be released with the specification describing these details. And of course, people can go to Terry and Bruce and ask questions about how to submit their prediction. I hope we get some predictions. I think it's a really interesting problem, and we're putting a lot of effort in, and we'll see how many CASP predictors want to try this one. Okay, so stop here, and the next meeting is going to be the first week in July. I think it's July 3rd. Okay, thank you, everyone.